Hello everybody, welcome to YouTube channel Unricher Biology. Today we will do reproduction in plants. What is reproduction? Reproduction is production of offspring of its own kind by any individual, by any organism, whether it is animal, whether it is plant. That is called as the reproduction. Human being will produce babies like human beings. Similarly, cats will produce cats, tiger will produce tigers. So this is called as the reproduction. That's multiplication of the organism, in which the to say by any method, that is called as the reproduction. Multiplication is its own kind of organism. So then in plant reproduction, it is of two types: asexual reproduction and sexual reproduction. In asexual reproduction, the sexual organs, sexual parts, reproductive part, they don't take part whether the reproduction is done by other than reproductive part. That is, it is maybe by stem, by the birds, by the leaf, by the roots. So when these parts, they help in propagating the plant, they help in producing other plants, then it is called as the asexual reproduction. Just like corms in colocasia, corms, they help in propagation of the plant, they help in producing the another plant. It also help in pollination, as in case of colocasia. Similarly, in case of onion, in case of garlic, these to say they also reproduce by vegetative organs. That is, in this case, the bulbils, they take part in the reproduction. Then you can say the scale leaves, you can say they take part in the reproduction. Then in case of ginger, rhizome also propagate the plant. It produces the plants by having the buds which produce to form the new plant. They also help in pollination, as in case of ginger. Similarly, in case of potato is the edible part of the potato which we eat, fleshy part, that is the stem, that is the modified stem. You might have seen there are eyes present on the potato. What are these eyes? Black spots, they are present on the potato. These are called as the eyes. These eyes are nothing, these are the buds. When you keep potato for some days, you will see that it will sprout at the points of eyes, at the points of buds. And if you sow these buds, cut the potato into the pieces which having these parts, these buds, these eyes, it will grow into another plant. This is propagation by the stem, modified stem. Similarly, bryophyllum leaf. Bryophyllum leaf, it is to say it, it is wavy, it has notches, and in the notches of the leaf, on the margin of the leaf, there are present buds which sprout on the new plant. This is also vegetative propagation. So in plants, asexual reproduction is also called as the vegetative reproduction. And you can say in some cases, lower plants, it is by spore formation. It is also asexual reproduction. And now we come to the sexual reproduction. Sexual reproduction, in which the reproductive parts, they take part in the reproduction. Reproductive organ of the plant is, we have done in the morphology, flower. And in the flower, there are 4 whorls are present. Outermost sepals, then petals, then stamens, and then carpels. Outer whorls, they are accessory parts. But the inner 2 whorls, stamens and carpels, they are the reproductive part. So first we will do the male reproductive part. Male reproductive organs. Which are the male reproductive organs? You can see Male reproductive part is, I told you, stamens. Stamens collectively they are called as the androecium. And each stamen has, to say, basal filament, and on the upper side, the filament is attached to bilobed structure which is called as the anther. The number of stamens vary from plant to plant. It might be single, then it is called as monoandrous. It might be 2, 3, 4, 5, more than 5. The condition is polyandrous. If they are free, then it is polyandrous. If the stamens are fused, then it is called as the synandrous condition. So what is the structure of this? By this filament, each stamen has filament and anther, bilobed anther at its top. By this filament is attached either to the thalamus of the, to say, flower, or it may be attached to the petals, then it is called as the epipetalous stamen. If it is attached to the perianth, then it is called as the epiphyllous stamen. This bilobe structure, anther, it further has two chambers. Each lobe has two chambers which are called as the theca. And if the two chambers are present in each lobe, the stamen is called as the dithecous. It is the most common structure, that is dithecous stamens are present. However, in some cases, no single chamber is present in each lobe, then it is called as the monothecous stamen. So, if what is the structure of anther? Sorry, it is called as the monothecous stamen, the dithecous stamen. And what is the basically structure of the anther? Anther, it has surrounded by, to say, three covering. Outermost covering is epidermis. Then next is the endothecium. Then middle layers, and innermost is the tapetum. You can say this is the epidermis. This is the endothecium. Middle layers, and innermost is the tapetum. If you cut the section of the anther, you will see it is just like circular and have four corners, roughly circular. It has four corners, and each corner there is present microsporangium. In each corner there is present a sporogenous tissue which form the microsporangia. So, and in the center, there is present connective tissue. This connective tissue joins the two lobes. This connective tissue joins the two lobes. So it has outer epidermis. You can see, if you see it in under microscope, this is the epidermis. This red one is the endothecium. These are the middle layers, and innermost, these nucleated cells, they are the tapetum. So each corner, it is if you cut the section, the four chambers are present. In each corner there are present, to say, microsporangia, and these microsporangia, they further produce the male gametophyte. So the outer three layers, that is epidermis, endothecium, and middle layer, they help in descent of the anther, and the innermost layer, tapetum, it is nourishing in nature. So cells of these sporogenous cells which are present in the corner in the microsporangium, they divide meiotically to form the tetraspore. Four spores are formed which are joined with each other. They are not separated. Their walls are separate. That is called as the microsporangium tetrad. And then this microsporangium tetrad, the process of, they are haploid, the cells are haploid because by meiosis, the microspore mother cells present here in the microsporangium, the sporogenous tissue, They divide meiotically and microspore tetrad is formed because the cells, four cells which are formed by the meiosis, they don't separate. They, they are retained as such. But when the conditions are dry, then what happens? They will lose water and microspores, they will get separated. And one microspore, from the one microspore mother cell, four microspores are formed. These microspores, they are called as the pollen grains. They are called as the pollen grains. They are, numerous cells are present in the sporogenic tissue. Microspore mother cells are numerous. They divide meiotically and each will form four pollen grains. Initially, no doubt, they are joined with each other to form the tetrad, but in the dry conditions, they will lose water after dehydration. Then these tetrads, they become separate and microspore, which are called as the pollen grains, they are shed. If you touch the flower of any plant, that is hibiscus, etc., or any other plant, some yellow-like substance will it will be touch your on your fingers. It what is that? That is nothing. They are the pollen grains. So pollen grains, they are each pollen grain. It is haploid. It is also called as the male gametophyte. Pollen grain is also called as male gametophyte. Each pollen grain which is covered by outer two membrane, outer two covering, outer two walls. Outermost is called as the exine and inner is called as the intine. So each pollen grain is covered by two walls. Exine, outer, and inner is the intine. Exine is very hard. It is formed of sporopollenin. If sporopollen is the hardest substance of the world, so this sporopollenin, it does not no enzyme can act upon it, no heat and high temperature they don't act upon it, and this is the hardest substance. This sporopollenin is present at one point at the tip. It is present at the one point at the tip, and how intine is formed of cellulose and pectin. Intine is formed of cellulose and it is continuous layer. Then in each pollen grain there is present a nucleus. The cell of the pollen grain will divide mitotically to form the two cells. One is the larger cell, that is called as the vegetative cell, and other is the small spindle-shaped cell, which is called as generative cell. Then what is the chromosomal number ploidy of this vegetative cell and generative cell? It is naturally haploid because they are formed from the haploid pollen grain by division, by mitotic division. So most of the pollen grain, they are shed at the two cell stage. One is large vegetative cell and smaller spindle shaped cell which float in the pollen grain, which is called as the generative cell. However, in other cases, in some other plants, this generative cell again divides into two cells and the pollen grain is at 3 cell stage, and it is towards the point where exine is not present and pollen tube is formed, and these two cells, generative cells, they are the male gametes. They enter the pollen tube and the pollen grains, they are shed in this case 3 cell stage. So in some cases the pollen grain, they are shed at 2 cell stage, generative cell and vegetative cell. Generative cell towards the point where there is exine is lacking, and in other cases generative cell divides mitotically into two. That is they form the two cells which are called as the male gametes. So they are shed in the 3 cell stage. So this is all about the structure of the male reproductive part. So what is their function? Pollen grain, they are to be shed and they are to be carried to the female part of the plant, female flower, that is stigma, by pollination, that we will do later on. This is the TS section shown under the, to say, TS section of the anther shown under microscope, outermost epithecium, endothecium, and you can say then these are the middle layers. Then this is sporogenous tissue which is forming the microspore mother cells. They are forming the pollen grains. Then we come to the female reproductive part. Female reproductive part of the plant, they are the carpels. They are the pistils. They are also called as pistils. In many plants, single carpel is present. Then it is called as the monocarpellary. In other plants, two carpel, bicarpellary, three carpel, tricarpellary, pentacarpellary, tetracarpellary, polycarpellary condition may be there. When the more than one carpel is present, the carpels may be free. Then condition is called as the polycarpellary apocarpus. If it is bicarpellary, then bicarpellary apocarpus, tricarpellary apocarpus. But when the carpels they are fused, then the condition is called as the polycarpellary syncarpus. Syn means fused. So then what is the structure of carpel we have done already in the morphology of flower. Each carpel has three parts: basal ovary, and then stalk-like style. Then is the receptive top structure, that is disc-like structure, is the stigma. That is called as the carpel. What is structure of carpel? So each carpel has three parts: basal ovary attached to the thalamus, then stalk-like style. It carries at the top disc-like stigma. The function of the stigma is receptive. It receives the pollen grains. And the style carries the stigma upward. Function of the ovary is to produce the ovules, which further converted into, by fertilization, they are converted into the seeds. So in the ovary, I told you that there are present ovules. In the ovary, there are present placenta also. It is a connect tissue. And to this tissue are attached the ovules. Ovary has chamber. It may be unilocular, it may be bilocular, it may be multilocular. In the chamber, there are present placenta. To the placenta are attached the ovules. So, what is the structure of ovule? Ovules, they produce the female gametophyte or the female gametes. Each ovule it is attached to the placenta by stalk-like structure, which is called as the funicle. This stalk is called as the funicle. And the point where this stalk is fused with the wall of the ovule, that is called as the hilum. That is called as hilum. Ovule has two ends, differentiated two ends. One end which is towards the stalk is the micropylar end, where there is an opening which is called as the micropyle. Opposite end is the broad end which is called as the chaliza. It is the chaliza end. Ovule from the outside, ovule has two covering: outer integument and inner integument. Outer integument and inner integument. Then these integument enclose a cellular mass which is called as the nucellus. Which is called as the nucellus, and in the nucellus is present female gametophyte, or you can say embryo sac. Have you followed my point? I'm to show ovule has two ends: micropylar end where there is opening is present, chalical end which is broad. It is attached to the placenta by stalk which is called as the funicle, where the stalk of the ovule is fused with the integument of the ovule, that is called as the hilum. And the ovule has two covering, outer integument and inner integument, which further they are converted into the seed coats. And these outer and inner integument, they enclose cellular mass, and in the cellular mass is present embryo sac or the female gametocyte. All these structures, except the embryo sac, they are the, diploid. So how embryo sac is formed? What happened? The cells of the nucellus, some of the only single cell in single ovule, it is functional. It is called as the megaspore mother cell. This megaspore mother cell divides meiotically. So 4 megaspores are formed. So what is the pleidy of megaspore? That is haploid. What is the pleidy of megaspore mother cell? That is diploid. So 4 megaspores are formed. Out of these 4, 3 they are non-functional. They degenerate. Only 1 megaspore is functional. That megaspore is called as the female gametocyte and phyte, and it develops the female gametes. How? What happened? The nucleus, this megaspore, it is also called as the embryo sac. It is also called as the female gametophyte. And the nucleus, this cell will divide. Megaspore will divide by mitosis. Consequently, by 3 mitotic division will occur. By first mitotic division, 2 nuclei will be formed. By second mitotic division, 4 nuclei will be formed. By third mitotic division, 8 nuclei will be formed, but no wall formation. Mitosis occur, but there is no wall formation. Cells, they don't separate. Only nuclei, they divide. Cell wall does not divide. Do you follow my point? I'm to say, megaspore cell, divide mitotically without the wall formation. It divides thrice and 8 nuclei structure is formed. And this structure, out of these 8 nuclei, 3 migrate towards the micropylar end, 3 migrate towards the chasal end, and cell wall appear among them. And 2 nuclei will be present in the center. These are called as the polar nuclei. Have you followed my point? I'm to say 3 nuclei, they are present towards the micropylar end, 3 towards the chasal end, and 2 nuclei in the center, which is called as the polar nuclei. And the micropylar nuclei as well as chasal nuclei, they will acquire the cell wall and cells are formed. So towards the micropyle, the cells, 2 cells, they are called as the synergids, and the central cell is called as the egg. So this whole structure is called as the egg apparatus. From the synergid filament-like structures, they arise. They to show the passage to the pollen tube to enter inside they guide the pollen tube to enter inside the female gametophyte, to enter inside the, this embryo sac. Then the 3 nuclei which are present chasal end, they also acquire the wall, and they are called as the antipodal cells. And the 2 nuclei in the center, they are present, and they are called as the polar nuclei. So now embryo sac has how many cells? 7 cells, but 8 nuclei. 3 egg apparatus cells in the egg apparatus, synergids in the egg, and 3 in the chasal site antipodal cell. 1 is the large cell, and in which 2 polar nuclei are present. So embryo sac has 7 cell and 8 nuclei. This is all about the structure of female gametophyte. Then we will do pollination and fertilization in the next step. I hope you might have followed it, and share it with your friends, subscribe my channel, like it. Okay, bye. Have a good day.